Let's move on to the process of photosynthesis. This is the equation of photosynthesis. Carbon dioxide adding with water with light captured by chlorophyll goes to carbohydrates and oxygen. In fact, photosynthesis is not a single-step reaction. It involves many reactions, each of which is controlled by different enzymes. The reactions occur in two main stages: photochemical reactions and carbon fixation. The photochemical reactions. Photochemical reactions occur in the thylakoids of chloroplasts. The reaction requires light. Therefore, they are also called light reactions. They involve three main processes: light absorption, synthesis of ATP, and the photolysis of water. First, light absorption. When the chlorophyll molecule in the thylakoid captures light energy, some electrons of the chlorophyll molecule are raised to a higher energy level and are emitted from the chlorophyll molecule. These excited electrons then pass through a series of electron carriers of decreasing energy levels in the thylakoid membrane. Energy is released in a stepwise manner. The series of electron carriers is called the electron transport chain. Secondly, the synthesis of ATP. Some of the energy released by the electrons in the electron transport chain is used to form ATP, adenosine triphosphate, by combining phosphate with ADP, adenosine diphosphate. This process is called photophosphorylation. In this way, light energy captured by the chlorophyll molecule is converted to chemical energy stored in ATP. The ATP will provide energy to drive the reactions in carbon fixation. Phosphorylation means the addition of a phosphate to a molecule. In photochemical reactions, the energy for phosphorylation of ADP is derived from light. The process is therefore called photophosphorylation. Then we move on to photolysis of water. The light energy captured by chlorophyll molecules is also used to split water molecules to form hydrogen and oxygen. This process is called photolysis of water. Hydrogen is then accepted by a chemical called NADP, nicotinamide adenine dinucleotide phosphate, to form NADPH. NADPH is the reduced form of NADP. It will act as a reducing agent by donating hydrogen in carbon fixation. Therefore, oxygen is a byproduct in the reaction. It is released as a gas into the atmosphere. A reducing agent refers to a substance that reduces another substance in a redox reaction. Reduction refers to the removal of oxygen or the addition of hydrogen. The hydrogen released from water is unstable by itself. It has to attach to a hydrogen carrier. NADP acts as a hydrogen carrier because the nicotinamide part can combine reversibly with hydrogen. When hydrogen is removed from NADPH, energy is released. So, in summary, the light energy captured by chlorophyll molecules has two main roles: synthesizing ATP from ADP and a phosphate through photophosphorylation, and splitting water molecules by photolysis to release hydrogen, which reduces NADP to NADPH. The ATP and NADPH formed will be used to drive the reactions in the next stage of photosynthesis, carbon fixation. Oxygen is released as a byproduct of the photochemical reactions. Now, let's talk about the next stage of photosynthesis, carbon fixation and the Kelvin cycle. ATP and NADPH formed in the photochemical reactions are used to fix carbon into organic compounds. This stage is called carbon fixation. It occurs in the stroma of chloroplasts. It is also called dark reactions because it does not require light. Carbon fixation involves a cyclic series of reactions. The reactions were discovered by Melvin Kelvin and his co-workers in the 1940s. Therefore, this stage is also known as the Kelvin cycle. It occurs in three main steps. First, carbon dioxide fixation and formation of a free C compound. Secondly, reduction of the free C compound and formation of glucose. And third, the regeneration of carbon dioxide acceptor. Carbon dioxide fixation and the formation of a free C compound. Carbon dioxide diffuses from the air spaces in leaves into the photosynthetic cell, and then into the stroma of the chloroplasts. Under the action of enzymes, a molecule of carbon dioxide combines with a molecule of a 5-carbon 5C compound to form 2 molecules of a 3C compound. In other words, the 5C compound acts as a carbon dioxide acceptor to fix the carbon dioxide into an organic compound, which starts the reactions in the cycle. Therefore, carbon fixation refers to the incorporation of carbon into the Kelvin cycle, while carbon dioxide fixation refers to the carbon dioxide being accepted by the 5C compound. The reduction of 3C compound and formation of glucose. Using energy from ATP and hydrogen from NADPH produced in the photochemical reactions, the 3C compound is reduced to another 3C compound called triophosphate. ADP and NADP are formed in the process. They will be reused in photochemical reactions. Triophosphate is a sugar. It is the primary product of the Kelvin cycle. 2 molecules of triophosphate combine to form 1 molecule of glucose. NADPH acts as a reducing agent. It loses hydrogen, which reduces the 3C compound to triophosphate. In this way, NADPH is oxidized to NADP. The regeneration of carbon dioxide acceptor. Not all triophosphate molecules are used to form glucose. Most are used to regenerate the original 5C carbon dioxide acceptor so that the cycle can repeat itself to form more glucose. The regeneration of carbon dioxide acceptor requires energy from ATP. In summary, the Kelvin cycle involves the reduction of carbon dioxide into glucose, using energy stored in ATP and hydrogen from NADPH. Now, let's talk about a common misconception. The misconception is, light reactions of photosynthesis takes place in the daytime only, and dark reactions take place at night only. The correct concept is, light reactions of photosynthesis require light. They take place in the daytime only. Dark reactions do not require light. They can take place both in the daytime and at night when both NADPH and ATP become available. So now, let's talk about the overall process of photosynthesis. Here shows the overall process of photosynthesis occurring in a chloroplast. Photochemical reactions and the Kelvin cycle are linked by ATP and NADPH. The equation of the overall process of photosynthesis is shown here. It shows that 6 molecules of carbon dioxide and 6 molecules of water are required to form 1 molecule of glucose. The hydrogen and oxygen atoms in water molecules are found in the glucose and oxygen gas produced respectively. The fate of the atoms in the substrates of photosynthesis is summarized here. Both carbons and oxygen form glucose, while the hydrogen of glucose comes from water. The remaining oxygens in water are released as a byproduct. Therefore, factors affecting the rate of photosynthesis. Here, we will study how light intensity and carbon dioxide concentration affects the rate of photosynthesis. The effect of light intensity. Hydrilla is used to study the effect of light intensity on the rate of photosynthesis. The rates can be determined from the amount of oxygen produced per unit time. First, we set up the apparatus as shown. Place the bench lamp at 0.1 meters from the hydrilla. Turn on the bench lamp and allow the plant to equilibrate for 5 minutes. Secondly, suck up the solution from the tube until the meniscus lies in the upper part of the pipette. Close the clip. Record the starting position of the meniscus. Third, after 5 minutes, record the final position of the meniscus. Record 2 more readings. Fourth, repeat steps 1 to 3 with the bench lamp placed at 0.2 meters, 0.3 meters, 0.4 meters, and 0.5 meters from the plant. Last, calculate the rate of photosynthesis in different light intensities. Therefore, at low to moderate levels of light intensity, the rate of photosynthesis increases. It increases proportionally with increasing light intensity. This is because more energy is provided to the plant to carry out photosynthesis. When light intensity has reached a certain level, the rate of photosynthesis does not increase with further increase in light intensity. This is because other factors, such as carbon dioxide concentration, become limiting. Here shows the effect of light intensity on the rate of photosynthesis. Initially, the rate of photosynthesis increases proportionally with light intensity as more energy is supplied to the photochemical reactions. As the light intensity increases, the rate levels off when the light intensity reaches a saturation point. The rate is now limited by other factors such as carbon dioxide concentration and temperature. The light intensity at the saturation point is called optimum light intensity. At very high light intensities, chlorophyll may be damaged and the rate of photosynthesis drops. The effect of carbon dioxide concentration on photosynthesis. Here shows the effect of carbon dioxide concentration on the rate of photosynthesis. Initially, the rate of photosynthesis increases proportionally with carbon dioxide concentration because more substrates are supplied to the Kelvin cycle. As the carbon dioxide concentration further increases, the rate levels off because the rate becomes limited by other factors such as light intensity or temperature. The atmospheric carbon dioxide concentration is 0.03% to 0.04%. Increasing the carbon dioxide concentration above this level can increase the rate of photosynthesis in most plants, provided that other factors are not limiting. Now, let's talk about the effect of temperature on the rate of photosynthesis. Photosynthesis involves a series of reactions catalyzed by enzymes. At low temperatures, enzyme activities are slow, and the rate of photosynthesis is low. As temperature increases, the rate of photosynthesis increases until it reaches a maximum at a certain temperature. This is the optimum temperature for photosynthesis. Above this temperature, the enzymes become denatured. Membranes of chloroplasts are destroyed. The stromata are also closed. As a result, the rate of photosynthesis drops rapidly. Here, we see a graph of the effects of temperature in the rates of photosynthesis. When temperature increases from 0 degrees to 30 degrees, the rate of photosynthesis increases. However, further increase in temperature from 30 degrees onwards causes a rapid decline, and eventually it will completely stop, even if there is further increase in temperature. So, how do we regulate the rates of photosynthesis? As the rate of photosynthesis increases, more food is produced by plants and the plant grows faster. Farmers can build greenhouses to monitor environmental factors such as temperature, light intensity, and carbon dioxide concentration, so as to regulate the rates of photosynthesis of their crops. In this way, the productivity and the quality of the crops can be improved. Greenhouses are often equipped with artificial lighting systems, carbon dioxide emission systems, and heaters. The light intensity, carbon dioxide concentration, and temperature can be increased to allow crops to reach their maximum rate of photosynthesis. The protected environment in greenhouses also reduces damages by pests, pollution, and bad weather. This is the end of the video. Thank you for watching. Please give us a like and subscribe to our channel. Click on the tab to watch the next part of the video series.